it could be. Okay, so we are recording for facilitated study group for blended micro on 11:30. We're going to be doing chapter 6 review and then doing a little preparation for the lab exam number 2. I wonder if Miss Aaron's working today.
She is. I just messaged her. She works in a Christmas store.
I know. Yeah, I
So
imagine they're crazy.
she may
Crazy
not
busy.
even be able to touch on this today.
Yeah, I imagine they're pretty busy. Okay, so we're talking about virology. And is it going to let me advance? There we go. All right. Viruses are obligate intracellular parasites. What does that mean?
They have to live inside a cell to
Right.
replicate.
Right. Yeah, the word obligate means they're obligated to, right? They have to. There is no choice. They don't have another way of acquiring resources or reproducing. They have to be intracellularly located in order to survive. Or they need a living host, right? That would be sufficient. What is latency? Think about like when she showed the growth curve for bacteria, there was a latent stage. What's going on during the latent stage?
I always think of it as like a late response, like a latency, like a oh my gosh, what's wrong with my brain right now? Like it doesn't happen right away.
Sort of a delayed response. Yeah.
Delayed. That's the word I was
Delayed or
looking
late.
for.
Yeah. Yeah. It's the time during which they're kind of like gearing up to do their thing, that sort of a period of dormancy, I guess you could say.
Mm-hmm.
Yeah, virus infects host cell and remains dormant. So it's sitting in there chilling, getting ready to create, an infection or to invade or whatever it's going to do.
Can we just discuss really quickly the cell structures that viruses lack, which cell structures, which
Okay.
categorize them as non-living?
Okay. Okay. Justina, you want to weigh in on that? What we did the other day in class, we talked about structures that viruses have that are like living things because they're not considered living. And then what features don't they have that would be required if they were to be considered living? So what characteristics do they have that are those of living things, first of all?
I just remember that they don't have metabolism, which makes them not like living.
Okay.
And as far as living, they're able to reproduce.
Okay. And what else do they have? What's their makeup? Like what structures do they have, or what's their
There's like a
physiological makeup?
nucleic acid.
Yeah, they have a nucleic acid. So that's a feature of living things, right? A genetic code. They have either RNA or DNA.
But which ones do they oh, I
So
remember.
they have DNA, and they can replicate. Those are really the only two things they can do that are features of living things. They don't have a metabolism. They don't have membrane bound organelles, or they're not very complicated structurally. Basically that, they require the host to as a source. They don't have a source of energy or utilize energy by themselves. They need the host to do that. That would be sufficient. Those would be sufficient for that kind of answer. Is there something else you were thinking of, Nicole, or?
The 3 blank spots on my page, and I remember us talking about it in class,
Okay.
but I
Is it in your outline? Where's,
yeah,
okay.
yeah, it says viruses. It's on the first page, close,
Okay.
almost at it's the second portion of the first page.
Okay,
Not portion, but the second little
where it says they
cluster.
lack which cell structures?
Yes. Oh, okay, so it's
What
actually the
she had in her PowerPoint was, well, what I have on here is nucleus, chromosomes, mitochondria. Now they have DNA, but it doesn't get to the point where it's, going through cell division like, eukaryotic cells would. So that's what I had. Justina, what do you have on your outline at that part? It would be the third. It says viruses lack which cell structures? I must have
because
gotten.
I put no organelles, but I'm like,
Yeah.
is there more than that?
Do one of you want to pull up the PowerPoint and see if we're missing something? Because I have nucleus, chromosomes, mitochondria, which are all cell structures.
I think that's probably right.
You're lacking
Actually.
those, and I think I got that from her PowerPoint. But if one of you want to pull the PowerPoint up just to verify, that would be good. We'll go on and we can come back to that. How's that sound?
I think that sounds great.
Yeah, so latent just means it's, give an example. Okay, give an example. I always think about the shingles and chickenpox. I couldn't remember chickenpox for a second. The varicella, because once you have chickenpox, it's sort of lying in wait, which is really what latency is. It's sort of lying in wait until the conditions are right, and if they are, that's when shingles can result. That's why if someone has had chickenpox, they should, as they get older, probably 60 plus, make sure they have the shingles vaccine, because it can be very dangerous for an older person, to have shingles. Does shingles show up? Let's say somebody hasn't had the chickenpox, and they're exposed to somebody that has shingles. What will happen? Will they get shingles or will they get chickenpox?
I don't know.
You have a guess?
You mean if they've never had shingles or chickenpox and they're exposed to somebody who has shingles?
Yes.
Um,
You got a 50-50 chance.
yeah, I mean, would it be like smallpox and cowpox? Like, would they just be immune to it then if they don't?
They wouldn't be immune to it. They would, but it would show up the first infection's going to be chickenpox. So since they had never had it before,
Okay.
yeah.
So if they're exposed to shingles and they've never had chickenpox before, they'll wind up with chickenpox.
Yes, potentially.
Okay.
If they catch something, so to speak,
Right.
it'll be in the form of chickenpox, which I think is interesting. Yeah.
That's very interesting.
Two main components of a virus's structure. Well, you guys said one of them a little while ago. You said DNA or RNA. What's the other one? What's the outer structure called?
The capsid?
The capsid, good. So DNA or RNA and the capsid. Or you can just say nucleic acid. Either one, you could say either DNA or RNA or just nucleic acid. What are viral spikes used for?
Justina, do you want to chime in too? Because I don't want to just.
It's fine. I'm actually walking, so
Oh, are you?
yeah, but the answer is adsorption.
I
Right.
don't want to like talk over you if you're trying to speak
She's
up at
good.
all.
No, you're okay.
Yeah,
It's really loud.
they're kind of an attachment structure. You know, they allow it to, it's kind of like the what were those other structures we talked about earlier that were for, oh, the fimbriae. They sort of have the same purpose. It allows them to attach. Yeah. Besides antivirals, how can viral infections be treated?
I mean, there's bacteriophages, but aren't those considered
Yeah.
antivirals?
Uh, no, they're a separate category, so that would be a good answer. Those fascinate me. I mean, it's amazing to me to think that, well, let me think about this a second. They're actually used, well, in that case, you're treating the bacterial infection. So that probably isn't the best answer for how viral infections, although it is in their outline under that, but the virus is actually infecting the bacteria. So the bacteriophage is used to keep the bacterial levels under control, which is really weird, I think. And there was an article somewhere, I don't know if it's the article she had you reading for that thing, supplemental thing we didn't get to, but they're actually used even in like produce to help keep, like, it's like a bacteriophage spray that keeps the bacterial level under control so you don't get spoilage in the food product, usually produce. I thought that was weird. It's like, well, oh great, so would we rather have a bacterial infection or a viral infection? But the viruses that they use, remember how they're very specific to a given host, so the viruses that are used wouldn't be damaging to the fruit or to the human. I think it's just amazing. Like, who figures this stuff out, right? Let's see what she said. Well, are there others? What others would you mention?
So in my notes, I've antivirals, gamma globulin,
Okay.
and interferon.
Yes, I think that's what she says here. Yeah.
Where is number 2?
And remember, interferons are what, oh, here comes Dembo. Okay. Are what, kind of promote white blood cell. Well, it would promote the white blood cell activity, I guess you would say. Let me let Dembo in.
Are we talking about the gamma globulin
I was talking about the interferon.
or the
Let's see. Gamma globulins, those would have to do with the antibody response, right? I think.
Yeah.
A
Immunoglobulin G.
response to an antigen, I think. Good morning, Mr. Dembo.
Hello, how are you guys doing?
We're good. How you doing?
I'm all right. It's in the final stretch of the semester.
Yes, the end of the semester is looming. I said to Nicole, at least be glad you're not her A&P class, because they have like 4 tests in the last, from Sunday to Thursday in finals week, because their lab exam is that week
Yeah,
and their lecture exam. Well, you guys probably have 3 though, don't you? Come to think of it. Well, no, you won't have a lab. You won't have a lab test, a lab quest. You'll have the final and the final lecture.
yeah, I have no idea I'm just hoping when I get reminded when it gets close.
Okay. Just look at that syllabus. I can help. We can look at that and make sure you like know
Okay,
what's
good.
happening.
But
Because
yeah,
I get confused too, and I don't have to do it. So
yeah, in my A&P 2 class as well, we have a couple
yeah.
of
Oh my goodness. Yeah, you got all of that.
exams. Like I think I have one today,
Yeah,
and
I
another
don't know
one
how you
next week.
possibly do that.
Yeah, luckily
Don't-
that one is not as I feel like the exams in that one are
Not
not
quite as
are not as
intense.
bad.
Yeah. Who do you have for,
I have Professor Hossam Farag. I don't know if you
okay,
know.
I'm not familiar with, is
Yeah,
that a him or a her?
yeah, he's a him. I'm a big fan.
I'm not familiar with him.
He's good. Yeah, he's.
Okay.
I enjoy his class very much.
Good.
Is very lecture focused, meaning
Mhm.
like what we actually do, less note
Okay,
intensive, which is nice.
okay.
Less notes. If you pay attention, you're focused, you engage in the class, you really
Mhm.
get more out of it,
Good.
than having to go and like study outside of the class. So I kind of like that.
That's your thing.
Yes. Yes. So what do you guys
We're doing the review for chapter 6, the review PowerPoint. And I'm trying to advance my slide at the moment. There it goes. How do viruses replicate?
uh well I think mitosis and meiosis?
Do they?
They have to enter the cell. They have to
Oh,
Yeah,
enter a human
they
cell
don't,
first.
remember, they don't have chromosomes, right?
right.
They
So
have
it's not
DNA,
mitosis.
but they don't have chromosome. They can't self they can't self-replicate really. They still
Oh,
need the resources.
gotcha.
Let's see what she says. Hijacked host cell metabolism, yeah. Um, yeah, so the materials that are needed, they have to acquire. They don't have them intact within themselves. There's been an age-old argument over whether viruses should be considered living or not, but since they're not independent, they can't be, even though they have some of the structures that are typical of living things, they just can't, they don't meet all of them. So that therefore they're still considered non-living. Kind of weird. What's a prion?
So it's a protein. There is non-living protein
Mhm.
that is
Yeah, it's just a piece of
hard to
protein.
kill.
And yeah, you think about it's just a protein. It doesn't have any of these other features that we're talking about that makes something viable, but yet it's very destructive. And yeah, the next question I think is about treatment. We'll get to that in a second. No, the next question is not about treatment. What's the cytopathic effect?
Real quick before we move on from that, how do prions cause damage to the human cells?
Anybody want to address that?
I think it's
Anybody
by
know?
altering like the way plasmids work, similarly to plasmids, like the protein will change the human cell protein, and then it will duplicate itself and create chains of itself, and that ends up messing up the cell.
Okay.
Okay, so it actually, like, in a sense, denatures the proteins in the human cells.
That's my understanding.
Okay. Thank you.
I can't
I don't know if that's right.
I can't really add anything more to that.
Oh, okay.
Is it treatable?
Nope.
No.
No, they're fatal. What would be an example of a prion infection?
Mad
Mad
cow.
cow
Yeah,
Yep.
chronic wasting, mad cow. Okay. Okay, cytopathic effects.
Then like the cells change shape based on the viral presence.
Yeah, it's basically, yeah, the virally infected human cells will change shape. And what do you call the open spaces that result? Like if the cells are kind of morphed and then there's open spaces, what are those called?
Not something into a plaque, but
Turns into a plaque, yeah. So the cytopathic effect causes the host cell to change shape. And then as a result, these sort of spaces will occur, called plaques. How are viral infections most commonly diagnosed? This one's almost like a trick question, I think. Like, do your thoughts would go to, is it usually a blood sample, a urine sample, like some of the other pathogens? You think about the specimens that you would take. Is that typically what they do for viral infections?
I mean, usually when I've ever encountered a doctor during such a time and they said, well, we think maybe it's viral, it's kind of like a process of
Right.
elimination.
Yeah. Like typically it's just based on the what's the
Symptoms.
the symptoms. So those things, if it doesn't indicate something else, then it's usually a virus, like you said. And they tend to run, like there's something going around, you always hear, right?
Yeah.
Something going around. Oftentimes the something going around is a virus, and you're not going to treat them with antibiotics because antibiotics aren't effective on them, right? So the antivirals and some of those other things, if they're severe enough, usually they let them run their course, right? There's something going around and we got to let it run its course. I can't begin to tell you how many times I've heard that in my life. Okay, we did that already. Chronic wasting, mad cow. I don't know what scrappy or scrapey is. Do you guys know what that is?
I can't remember.
Yeah, I don't know what that is. But you can use mad cow or chronic waste, CWD, chronic wasting, which is usually in deer. Typical size of a virus, is it bigger or smaller than a bacteria?
Smaller.
Smaller. It's funny because Nicole, you mouthed it, but Dembo's voice came out. Like
That's so funny.
that was really funny.
Smaller.
Smaller. It was like you were voice throwing or something. That was a riot.
Oh no.
That was too funny. And it was like precisely timed. It was better than Cher lip syncing at did anybody watch the thing last night? The Rockefeller Center Christmas tree thing.
No.
Cher was on, but she was clearly lip syncing and
Oh,
not doing it
no.
very well. You were much better at lip syncing without even trying.
Wow.
That was
I missed that.
Yes, well, it was almost comical. I don't know. It was just funny. Anyway,
I hope
sorry.
it records that way. I hope it shows up.
Sorry. I don't know why things like that make me laugh, but anyway.
Well, because it's funny.
It is funny. It was just perfectly timed. Okay, so small, right? 0.2 to 0.4. Viruses are smaller. She likes the ordering things. We probably did that in the first couple of chapters, right? That would be a good thing for the final, like what is the correct, size orientation of different things like virus, bacteria, red blood cells, white blood cells, those types of questions. What is transduction? That term's popped up a few times through the course.
That's where the genetic information is transferred over to the host cell.
And what typically would do, like how would
bacteriophage.
Bacteriophage, right? Exchange of DNA between bacteriophage and bacterial cells. So the viral DNA is basically being transferred to the bacterial DNA, right?
Mm-hmm.
So the bacterial DNA, and that's how it's used. Bacteriophages can be either positive or negative. It depends on the application, but if they'll control the growth of the bacterial cell. So they may have their own, they may be infective themselves, but they also could be used as a control measure, which I think is just fascinating. It amazes me that we have figured those things out. And there's so many other things we haven't, but that we can use bacteriophages to control bacterial growth. Kind of crazy. I don't know if they're used in that reading thing that you guys had. I don't remember. Did it say anything about bacteriophages being used in like oil spills? You remember anything about that? Somewhere along the line, I read something about that. Like they can be used to help control spoilage in, like we were saying, in produce, in like the fresh fruit markets and places like that, in, production facilities that make fruit for, distribution into.
Yeah, I did read something about the fruits.
Yeah, I think
vegetables.
that's very interesting. But
Mhm.
because viruses are so specific, they won't infect the fruit and they won't infect the human that eats the fruit,
Right, just the bacteria,
just the bacteria.
which is nice. So that's kind of like a form of the phage theory thing that
Yeah, the
case study.
phage therapy, right. Mhm.
Yep.
It's just used on, for a different purpose rather than in the human. It's used on the food. Why do
And speaking of the phage theory case study, we were we read and we were going to talk about, but we ran out of time.
yes.
So the first question was like, why weren't they concerned about it sickening the patient?
Yeah.
Is it
Um,
is the answer to that as simple as, well, again, it only focuses on the bacteria inside
right.
the patient and
Exactly.
not the host cells,
Exactly.
non-bacterial
They're
host
going to
cells.
be specific to the bacterial cell. They're not going to see, they're seeing the bacteria as their host rather than the human cell.
Right. Now they would also attack the normal flora, correct?
Potentially. I don't know if they're specific to the point of only infecting pathogenic bacteria. I don't know. You know what I mean? I don't know if
Yeah,
they,
yeah.
if
It did say something about the particular bacteriophage used were specific to the pathogen that the
right. So in that case, then they wouldn't
yeah, I guess.
affect the normal flora, I would say.
Right.
Unless I don't know if it's as specific as this, down to the species level, or if it's like a certain genus. But let's say you have normal flora of staph, but you also have pathogenic staph. Will it affect the normal flora in that case? I don't know. I don't know the answer to that. But
Right.
But the same thing is the specificity aspect is why they
Yeah.
would not, in general, damage.
Right. They can
either
be
human.
useful in clearing out bacteria,
Right. It's amazing.
while
Really is amazing.
preserving the rest of the thing, the host.
Yeah, very
Yeah.
interesting. Why do traditional culture techniques not work for viruses?
Because they need a living host.
Yeah, so if you were trying to propagate or grow a virus, and we can't really even call them colonies, but a population of viruses in a lab, what would you do?
You need mice.
Yeah, usually mice or rats
Something living.
would be the typical hosts. Sometimes they use other things like rabbits and dogs, unfortunately, but yeah, you'd have to have the other thing they use a lot. Do you remember? Anybody ever remember? It's a product of an animal. Does anybody remember? She said it in the notes, I think.
Mm.
The embryonated eggs.
Oh
Yeah,
yeah.
embryonated
Yeah,
egg.
which are basically,
Mhm.
chickens in utero, not utero, but in vitro.
Now, are they talking about like any egg or like eggs that have like matured almost to the point of hatching?
Well, it would probably be an earlier stage. I mean, they don't have a real long, quote unquote, gestation. I don't know point of being fertilized to hatch, I'm not sure what that is, but it's not very long. So they would be fertilized eggs. I mean, embryonated would mean, that they would have been fertilized.
Yeah, they would have gone past the stage of the eggs that you normally eat.
Right. Yes, they'd be
So-
fertilized. Typically, the eggs we buy aren't fertilized. They're just laid,
Right.
they're eggs that have not, like the chicken hasn't even been present in close proximity
Yeah.
to a rooster.
And they probably also do something to those eggs to stop them from continuing to mature, right? Because if you buy an egg and you don't use it for like a long time, it does not eventually like
No, it'll
fertilize the young
never,
on the
no,
stage.
because it hasn't been fertilized. It'll just, it'll just get bad. It won't, but it won't, every egg doesn't eventually produce a chicken if you let it go long enough. It has to be a
No?
fertilized egg.
So if a chicken hatches an egg
Mhm.
and and let's say, the egg stays there with the, in the whatever, in the chicken coop,
Yeah.
there's a chance that that egg might not hatch.
It won't hatch unless it was fertilized. It's not going to become a chick unless it was fertilized by the male chicken or the
Hold on a
rooster.
second. Hold on.
Like it
Now
has to be
now
fertilized.
we're opening a whole Pandora's box about. So hold on, you're telling me if a chicken hatches an egg,
Mhm.
there's a stage there where it needs to be fertilized beyond it becoming an egg and out of the hen's
No, it's
body?
fertilized. It had to be fertilized before
Oh
she
yeah, no, that's what I'm talking
she
about. I'm talking about the point where it was actually hatched already.
No, they're fertilized outside. Like
What?
they're
After an
fertilized
egg is
after the leg.
No.
after an egg is hatched, it's fertilized after that?
No.
What are we talking about here?
What you talking
No.
about?
Aren't they fertilized after they're laid? Doesn't the female lay the egg and then the
No.
male fertilizes it? Or do
I
they
don't think so.
No,
actually
it's fertilized. It's fertilized internally.
Yeah.
The
Oh.
fertilization process takes place.
I thought they were fertilized. Like I thought that the male chicken like laid on top of it or like.
Oh no.
No.
A hen will never allow a male chicken anywhere near.
They
near
have
her
the
fetus.
same,
Yeah, I guess
basically
you're right.
they reproduce sexually like humans, like they would have not the same kind of parts, but the male will need to copulate with the female.
Yeah.
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 So which came first, the chicken or the egg sort of story? But yeah, unless the fertilization, unless that act took place and fertilization occurred, the egg, when it's laid, is just an egg. It would
It's
be
just
like
an egg that
an
just takes
egg that
time.
isn't fertilized, and it's just gonna sit there. It's not gonna eventually form into a chick because it's not a fertilized egg.
I didn't know that.
Hold
Now,
on.
sometimes, especially with free range chickens,
Yeah.
because you don't know where they are, or who they're hanging out with, right? Free range chickens sometimes, unbeknownst to the chicken farmer, or usually they're smaller farms type of thing, but sometimes they could be fertilized, and you'll get, if you crack an egg and it has like a little blood spot in the yolk, that often means that it was a fertilized egg. So that one, if it were incubated, I mean, by the time it's like in the chicken laid it, you collected it, and it's refrigerated, it's not gonna develop because it hasn't been incubated properly.
I guess that's what I'm asking.
Okay.
So because back home in Gambia, Africa, we would like we're not scientific in our chicken breeding there. So
Okay.
you have your chickens, you got
They're
hens,
just running around doing their thing,
you got the
doing what
roosters,
chickens
they do their
do.
thing, and then couple of months or whatever later, the hen starts laying eggs, And then you just let the hen do his thing, and then the eggs hatch, and then they got baby chicks running all over the place.
Yeah, well, but
It's-
she has to sit on them, you know, like she
Right,
has
yeah.
to
So
sit on them and hatch them. They have to go through an incubation period, which isn't real long. I don't remember what it is offhand. I should know, but I don't. But yeah, so if they're not incubated, then they won't have the proper conditions for developing. You know what I mean?
right.
But yeah, good question. Yeah, so there it's more just like free range type of thing. It's not controlled. You know, it's not the big mass production that we're doing here, right? You know,
Right.
here they're in hen houses, smashed like 5 or 6 of them to a cage, and they just keep cranking out eggs. But
Right. But so the
aren't
incubation
given the
period is
chance
what?
to be fertilized
Gotcha.
because they're not even in the same house with the males. Fun fact, I worked for a poultry veterinarian back, I had an internship in college, which, you know, like ancient history ago, but they keep like, we called on turkey farms like Round Hill and places like that. And they keep the males and females separate because, yeah, just what you were alluding to, the males will just be like, they'd be bruising the meat of the females because they want to mate with them. So they would keep the males in a separate house, and part of the job of this vet that I worked for was artificially inseminating chicken or turkeys. It was, and that is not a pleasant experience.
Oh my goodness.
We had these guys on work release from Lancaster County Prison. And so it's me and these dudes from Lancaster County Prison going out to these farms, and they would this is more information than you possibly wanted to know, but they would actually do what's called milking the turkeys. You'd flip them upside down and you're basically, you know,
Wow.
activating the turkeys.
Oh
That's
my.
crazy.
To milk the semen out of them, and they collect it in these test tubes. And then here are these big guys, these big, you know, prisoners on work release, trying to keep, because they had to keep it warm, because, you know, otherwise it would die. The turkey sperm cells would die off. And then they would have to take it to the hen house. And at the hen house, I was the pipetter. We would put it into this machine, and then they flipped the female turkeys. And a turkey that is being like turned on its head to be inseminated is not a happy turkey. Let me
No.
say that. So they're squawking and flapping their wings, and they're strong. You know, you figure a turkey that ends up being a 20-pound turkey on, you know, on your Thanksgiving table,
Right.
she's like a 35-pound turkey with all of her guts and feathers and parts intact.
Oh,
And she's like ticked
right.
off because we're trying to jam something up her hoo-ha, you know.
That's crazy.
So, oh, this is great. We're all recording all this. So anyway,
I love it.
but then they pipette the, you know, the turkey semen into the female, and then send her on her way. And then she's gonna lay, you know, eggs that are fertilized, and then they grow into the baby turkeys. Yeah.
Wow.
So there you go. Here's your turkey lesson of the day. But Nicole, a lot of animals, especially amphibians, will lay eggs and then they are fertilized outside of the body.
I
So
thought those were like that too.
yeah, you're not totally off base. There are
I
some
thought that
animals
was
that
just
do
like
that.
a bird thing.
No,
I thought that was like a bird and amphibian thing.
well, there might be some birds that do that, but chicken, I can't think of one offhand. I think most of them are, you know, basically what we're talking about, but there may be some that do that. But it is a method of reproduction that, you know,
Okay.
some frogs, salamanders,
I
stuff like
read that
too.
somewhere.
Yeah. So you're all right. It wasn't a crazy question. All right. That was fun. So embryonated eggs are one of the ways that they culture viruses. And they'll do them in a lab, a simulated lab environment. Like you can like cloning, that's another thing that they use embryonated eggs for. They implant the DNA, inject it into the egg, you know, in a lab setting. So that's a possibility too.
Like, that's a way to mass produce eggs without having to have them hatch?
Well, they'd still need to hatch. But they can be done in a simulated,
No,
lab
I mean like
simulated
not having to
environment.
like have
Not
them,
have the chicken sit on them. Yeah.
yeah, or like no, have them come out of a chicken, like literally.
Well, they would have had to come out of a chicken at some point.
Okay, it's not.
But then they can fertilize them in a lab setting. It's very strange. Yeah.
This
Oh.
might sound like a not so brilliant question, but like in vitro babies,
Mm-hmm.
is it possible for them to mature into full-grown babies without going through the womb?
Well, I'm sure that there's been some attempts at doing that, and we probably don't even want to know how far
Oh
along.
God,
I
definitely.
don't know that they've done that with, look, like cats and sheep and things like that. If it's ever been successfully done with humans, it's kind of one of those situations where we as the general public are not going to know it because it's a very controversial situation.
Right.
I the incubation period is also so much longer that I don't know. In theory it should be possible, but whether it has ever been done successfully,
Yeah, I'm sure it's definitely been attempted.
yeah, I would think so too. I've always picture all these like botched attempts at doing that, just like floating around in laboratory settings in big
Oh
vats
yeah,
formaldehyde.
I have a quite a vivid image of what it would look
Yes,
like. It is not
Yeah.
pleasant.
I do too. And it's in some like high security facility where
Yes,
no one ever,
you hear alarms going off.
yeah, it's
Beep,
very
beep.
dimly lighted,
Yes.
and
Yeah.
our imaginations are running a little wild. Oh, well, we talked about the plaques. What are plaques again? I always think of plaque being like the stuff that deposits into your blood vessels, but
Plaques. Aren't they
or on
like
your teeth.
biofilms?
Well, they are, but they're also, when it deals with viral infection, it's actually like a space the tissue is sort of like eroded away rather than a deposit of like what we're talking about with a biofilm like on the teeth or in the blood vessels. It's sort of a space that develops as a result of
Isn't that one of the
the
cytopathic?
cytopathic effect. Yeah.
So yeah, okay.
Cytopathic meaning pathology, pathology meaning disease of cells, cytopathology. And the plaque is like the spaces that form. Picture it like the virus kind of like almost eats it away. Like chronic wasting disease is an example. It literally is like wasting the tissue away.
Like
The
if
plaques
it,
would be like a minor form of that, I guess.
so it would like if I think of plaque in teeth, it's like like forming of like almost like biofilms.
Yeah, it's more of a
Clumping type
deposit
thing.
onto a tissue, right? Like,
Right.
and this is sort of the opposite because it's more of an eroding away, a space that forms. Yeah, I think a plaque is, the stuff on your teeth, the arteriosclerosis, if you have what they call hardening of your arteries, that the plaque is what's building up on the inside.
Right.
Whether it's lipids or other stuff. Large open spaces due to tissue destruction. Okay.
Yeah.
So yeah, kind of our English language, the word is the same, but it has a different meaning, right?
Mm-hmm.
What is the challenge in creating antiviral drugs? Think about COVID. What's a potential challenge?
It's really hard to do without hurting the tissues.
Okay, so it could have
With
detrimental
the
effect
cells.
on the host tissue. What also, the flu, COVID, there's a lot of different
Strains.
different strains. So it'd be hard, like the flu vaccine every year, they kind of have to project or predict what
Mhm.
is going to actually, what organisms are going to be causing the flu, right? They sort of have
Right.
to second guess, I guess, or pre-guess. Predict. I don't know if that, let's see what she says here. Must eliminate virus inside the host. Okay, that's what you said. But also the idea of the fact that they can very quickly adapt and morph and become a different strain, right? I would accept our answer. I don't know if she will. But there, you had the answer, Nicole, so you're good. How are prion infections treated? Well, we know that because we already talked about it. Are they?
No.
No, they're not, because there isn't a treatment and they're fatal. It's a bummer. That might be the last question. No? No? What must happen first for a retrovirus to replicate?
Mm.
It was changed from what, RNA to DNA.
Yes, and I always think about like retro is like a throwback, right? Like a retro.
Going backwards.
So it's going backwards. So it's reverse of which process?
Replication.
Yes, it's a reverse. Well, not replication. What's the
Oh,
one?
transcription.
Right, reverse transcription. So we're reversing. So we're going from RNA back to DNA, right?
Mm.
Kind of crazy. Reverse transcription, convert RNA to DNA. So think of the word retro. Retro is going to throw back to the origin, which is the DNA. How are viruses released from a host cell?
She's very, very busy. She's not, she just realized it's
Oh.
Thursday
Oh,
and she's very busy at work. So she
okay. Well,
probably
tell her
hasn't
we're
been able to-
recording, that I remembered to record for once. Tell her we missed her and we're recording. So how are they released? There's a couple different ways.
Exocytosis
Exocytosis.
and lysis.
Okay, good. Which one is destructive to the virus?
That will be lysis.
Lysis, because lysis means death, right? To lyse means to break apart.
Mm-hmm.
But whereas
Then-
exocytosis, that remember they kind of form that little vacuole thing and things are released, and then
And they actually take on the membrane of the host cell to form
Right.
envelopes as they come out,
that's right.
one by one.
Yep.
In lysis, they all come out at once.
Okay, good. Yeah, lysis is just like a like a burst of, I always think of like a seed pod on a plant or something. If you have rupture it, they just all scatter, like the spores all fly around or whatever. How are they taken in? How are viruses taken into a cell? What's that process called?
Endocytosis.
Endocytosis. Is there another one?
And fusion.
Yeah.
And
Penetration.
fusion. That's right. I don't know if she asked that.
Fusion. Okay.
Did she ask that question here? Oh yeah, that's the next question. Okay. All right. So endocytosis and fusion. Good.
What's the difference between the two?
Well, endocytosis works like exo, except it's going inward. So like a little vacuole forms and it brings it in. Remember the phagolysosome and all that
Yeah.
good stuff? It does that. Whereas fusion's kind of like the reverse of binary fission. Binary fission just splits.
Yeah.
Whereas fusion, it sort of just like gets incorporated into it.
So which
Sort of
one
merges with it.
which one involves the envelope on the virus? One of them,
Endocytosis. I
that's the
think.
one where the virus has the envelope.
That's a good question. Let's look it
I'm
up.
not sure if I'm completely following.
I would think,
Actually,
hmm,
let's talk about it instead of me talking about it because I'm just gonna look stupid.
no, don't say that. Go ahead, tell us what you know. Here,
Okay.
okay. The virus enters the host cell in two major ways. Endocytosis, the host cell forms a pocket and brings the virus in. In fusion, the virus fuses with the host cell, leaving behind the envelope and the viral spikes. So fusion would be, it kind of like sheds its outer, it like has like its little coat of armor on, and it throws it off so it can fuse. Kind of
So in
interesting.
endocytosis, the virus goes in with its envelope.
The host, well,
Right, like endocytosis, the virus goes in, it becomes like a sealed vacuole
well,
inside of
yeah, the host cell
Right.
itself is what makes the little, well, it's a vacuole, yeah. And it actually incorporates the virus into that.
Mm-hmm.
Right.
So whether or not, I would almost think that it wouldn't, if it had spikes or an envelope, that it probably wouldn't do that. Like I would think it would, I'm not positive about that, but I would think that those structures would prevent that from being able to happen. But I think if you know the one, if you know the two methods, and the endo involves the vacuole, and the fusion involves actually, joining the two together. You think about the word fuse to put together. But yeah, that's a good question, and I don't know the answer.
This is what I asked my AI, and it says
Okay.
when viruses enter the host cells, they can use different methods to get in. Fusion is when the viral envelope merges with the host cell membrane, allowing the virus to release its genetic material directly into the cell.
Okay.
Endocytosis, on the other hand, is when the virus is engulfed by the host cell, forming a vesicle that brings the virus inside the cell. Both
Okay.
methods help the virus gain entry, but they differ in how the virus interacts with the host cell
Okay.
membrane.
Good. And it doesn't really say whether that one, the endocytosis would incorporate it with its spikes or whatever. And
No,
it's
but
really-
it
very
sounds like the
interesting, but-
everything goes in with the endocytosis.
right.
And in
Yeah.
fusion, the envelope is incorporated with the cell membrane, and then it's just a naked virus going in.
Okay.
Just the capsid and the
And
nucleic
the
acid.
nucleic acid. Okay. Thank you for your AI.
Yeah, my AI is brilliant.
It's amazing, isn't it?
It has helped me so much in this class alone. Just anytime I come across questions like this that I can't find the answers to,
Yeah.
whatever, I just ask AI.
Yep.
Like
I
AI
don't know
can,
how we survived without it, to be honest.
I don't know how we're gonna survive without it going forward. It's gonna take over.
Yeah, it's a little scary, actually.
It
But
is. It is.
what is one care? And we already talked about these things. But go ahead. How are they? One carrot. Do you hear my dog howling again? It seems like she does that every week when you guys are on. It's so pathetic. Oh my gosh. So DNA, what else did we say? Or DNA or RNA, nucleic acid, and
Well, it is yeah, it has genetic
yeah, it has genetic information.
it reproduces,
It reproduces, yeah. It needs a host to be able to do it, but it does do it.
but it still does.
Yeah.
Yeah.
The active or inactive, I don't know. It doesn't, I would use one of the first two or both if she asked for two. But she said one.
I don't feel like virus is active or inactive really answers that question.
Yeah, because
I'm not sure why
volcano
you
can
would
be
agree.
active or inactive, and it's not. Yeah, I don't think that's a living thing necessarily, but okay. I like the reproduce and nucleic acid. And that's the end of that one. Okay. Let's, oh, and on the viruses, did you guys all see on the, she'll have a set of, I think they're multiple choice, like that will address several of the viruses. And at the bottom of the At the bottom of the study guide, the one that's a separate document from the outline, she has a list of the ones that you should be responsible for. Do you guys know where to find that?
No, but I definitely would like to
Yeah,
Yes.
because those are like, I mean, you probably know a lot of them. My problem is I know like the common name, but I don't necessarily know the scientific name of them.
where can you find the study guide?
It's right in the same area on D2L as the outline. There'll be like the PowerPoint, then the outline, and then the study guide, I think are all there
Is this
together.
for week
What
14?
are we in 13?
Yeah,
Or is this week 12 or
Yeah, I lose track.
13, I think. Hold on, let me double check, tell you.
I think
Yes,
week 13 for the quiz and the lecture.
okay, so it should be in, yeah, week 13.
Okay, study
D2L.
guide. Virology,
Yep, you're getting
chapter
very
6.
warm. Chapter 6, yep.
Okay, so yeah, it says specific viruses. The information is found in DNA/RNA virus PowerPoint.
Yeah, so
PPT.
those, know what those are. Like
So
it'll be a
rhinovirus, RSV,
yes.
Cox,
Coxsackie virus.
Coxsackie virus, Epstein-Barr virus, Varicella zoster virus. Like what does she want us to know? They're like
Well,
common names?
You should know, yeah, what it causes.
Oh, okay.
And, like as an example, Coxsackie virus, what the heck? I want to say that's the hand, foot, and mouth disease one, but I'm not positive about that. Let me see. But yeah, know what disease it causes, and a couple of symptoms, because she could say something like, let me find one. Like something like it's an abnormality in your white blood cells, it causes chronic fatigue, and overall sleepiness. There's both a viral and a bacterial form of this. A lot of times college students get it. It's mono, but it's Epstein-Barr virus, and I didn't know the Epstein-Barr part of it. I know what mono is. So you should know the infection and what of those that she has listed. Know the infection and then some symptoms. Like if someone presents with the symptoms, what is it likely to be? Because
Cool.
those are kind of like freebie points, but they can be destructive to your grade if you don't know them. You know what I mean? Like if she has 3 or 4 of those on your quiz, it can make or break your grade for that quiz.
By the way, do we have any more opportunity for extra credit?
I don't know the answer to that. Do you, Nicole?
I don't think so. There's nothing that's in the grades.
Yeah,
There is a section where both of our extra credit things are
posted, and they're just the 2 of them. There isn't
Yeah,
a third.
so
Okay.
I mean, I don't know if she's gonna add anything in for, people who come to study group or anything
You
like
might
that.
mention that.
Probably
You
won't, But
might mention that. Say, hey, how about a couple extra points for study group attendees, buddy? Like I send her the list every week of who's here. Like we have a running, like
that would
spreadsheet.
be so lovely if she would, but
Yeah.
I doubt if she
Hey,
probably
wouldn't it be
would.
nice if we would get a couple points for coming to study group?
That would be nice.
Oh,
I could
my.
really use them too, because these last 2 labs I did not do as well on them.
Oh.
So that would be lovely.
Okay, well, speaking of lab, we're past time as usual. We can go till 1:30. Is there anything on the lab lab materials for the big lab test. Well, wait, you don't have one. Do you have one this weekend? This weekend? No. Yes, no?
Probably.
I can't.
A lab
Do you have a
exam.
lab? Do you have a lab quest due Sunday? Or it's, I don't, or is it just
I
your
know
final?
we have our lab exam too that's due,
Yeah, you have your big lab exam. That's due by Sunday, right? Do you?
but
Hold on a second.
hold on, let me
This
double.
Sunday?
Oh, you do
Fine.
have a lab quiz. You have a lab quiz, the API lab. The API one. That's due on the 3rd. It's open now until the 3rd. And your big lab exam, yeah, Dembo, don't forget to do that because that's a lot of points.
Hold on. When is that due?
Oh no, it's due on Sunday. It's due the 3rd.
This Sunday?
Yes, sir. You got it. It
The
says
big lab exam.
Yes, the lab exam too. The one you
Oh,
reviewed for
Lab
with
exam
that
two and lab 12 quiz.
that
Oh,
one
thank
you,
God I came to this study.
I'm glad you did too. I'm sure you're not alone. Yeah, the review that you did the other day in class, that near
Yeah.
review, that was for the lab exam too, which is
Yeah, I knew that, but I didn't realize it was this
yes,
Sunday
it's
that's
due
due.
by Sunday, which is good and bad. It gets it out of the way, so then you can worry about your lecture exam. But yeah, it's coming up on it. So the
Okay.
question is, does the API thing I think makes good sense. Was there anything about that that was confusing for any of you? We'll talk about that one just briefly if we want to.
The lab, the API.
Why
Oh
do
yeah,
you
that's the.
add water to the tray?
Oh yeah, so it doesn't dry out.
Yeah,
during
to keep it
incubation.
hydrated. Why do you add oil to some of the little wells?
To eliminate oxygen, so it's an anaerobic reaction.
Right.
Yep.
Which group of organisms are API test strips used for? Gram positive or gram negative?
Gram negative.
Gram negative. What kind of tests are they? Biochemical.
Biochemical.
Yeah, and remember, biochemicals looking for either enzymes or
Uh, oh yeah. What do you call it?
not the reactants, but the
end products.
products. Yeah, so biochemical testing is looking for either enzymes or end products or both.
And these are McConkey like
Yes,
right?
because you're using
That's for
you're trying
differentiation.
yes, you would first
I mean, for
place them on
inhibition.
McConkey auger because and then you would introduce them to the test strips. You make like
So
an infusion of them. You dilute them in a certain amount of sterile water. I don't remember exactly how the infusion's made, but then that's added kind of drop wise to the little wells.
is the McConkey agar part of it? Is that because we want to inhibit the gram positive and just
Yes.
make it gram
That's
negative?
exactly right. Yep,
That's the reason why gram McConkey is used.
that's right. You got it. Yeah, you're encouraging the growth of the gram negative organism and inhibiting the gram positives. And what are the only two media that we talked about that are both selective and
Inhibitor.
well, selective and inhibitory,
Well,
remember, are the same thing, because if you're
differential.
selecting for something, you're inhibiting. Differential, like that word escaped me. Yeah,
Right.
there's only two. Which two are they, remember?
That will be McConkey and mannitol.
Yeah, the two M ones. The face-to-face class yesterday, they were having a hard time with all of that. What trying to think of anything else with the API test strips. I feel like
So everything else is
is
differential,
only
right?
differential. That's right. Now, it doesn't mean that there aren't some, but of the ones that we studied,
Right.
the only two that are both are McConkey and mannitol salt. What makes McConkey differential?
What's that thing called? Crystal violet and
That's the inhibitor. That's what makes it selective.
oh, okay. That'll be the this starch.
Well, it's not, it's a carbohydrate. It's not starch. So what carbohydrate is in the McConkey?
Oh, no, what am I? You see, these ones trick.
Yeah,
I
these are
always lose them.
tricky. That's why that little chart that we were working on, if you could, use that to help study, because I mean, when I made that up because it was like, I can't keep this stuff straight,
Right. So
It's
is
one of
the
the oses, which
yeah,
ose?
lactose.
Lactose, yes.
Ugh.
Lactose, you got it. What makes mannitol salt selective? And remember, selective and inhibitory.
Salt.
So if I ask
Mannitol.
that question, mannitol is what makes it differential.
That's salt.
Oh, the salt. Okay, good. Salt or NaCl, right? Because it's going to inhibit everything except what? What does mannitol salt grow?
Staph aureus.
Staph aureus, good. Well, staph, and then if it's positive as a mannitol metabolizer, then it's Staph aureus, right?
Right.
Yeah.
Okay, staph.
So staph at the selective part of it is
Right.
selecting for staph. The differential part is further differentiating and saying, okay, it's Staph aureus, right? What's the first test that you always do for gram-positive testing? After you've done that, you of course did the gram stain, and
That
that
would be the
it's
coagulase test.
not coagulase.
For gram positive?
For gram-positive, not coagulase. What's the one you do before coagulase? You're close. It's a C word.
Catalase?
Catalase. You do the
But
catalase.
it wouldn't that be gram negative?
No, that's the gram-positive. Gram negative, remember the biggest difference is gram negative, it doesn't matter what order you do things in. But gram positive, it's like a little flow chart. It matters that you do what you do first, because one leads to the next. So in other words, you do
Right.
catalase.
Oh, okay, gotcha.
If it's positive, then you do what? If it's positive,
Then if it's positive, then it's a staph. Then you do. Right. Now I'm confused.
okay.
I'm confusing myself.
If it's positive for catalase, it's a staph.
Yeah.
And then you do, what was the one you said? And I said, no, not yet.
Well, if it's staph, then you
Then you do the what test?
coagulase?
Coagulase. And
Right. Okay.
coagulase and mannitol salt are kind of confirming tests for each other, because they
Right.
both confirm Staph aureus, right?
You know what? You know why I'm confused? I was thinking bacitracin the whole time.
Okay, well
Okay,
thanks for the lead in. I was going there next. If it's catalase negative, then what do you do? What genus is it? It's not staph,
Bacitracin.
it's
Oh, that's strep.
strep. And you do the what test then?
Bacitracin.
Bacitracin. Let's say it's sensitive to bacitracin. Well, first of all, if it's sensitive to bacitracin, what's it going to look like? Are you going to have a zone or not have a zone?
You will have a zone of
If it's
inhibition.
sensitive, you'll have a zone. And what organism will it be?
That would be strep A.
Strep A. I always just remember strep A. Strep A is sensitive, if you can remember that, which is the strep throat organism. And then what would you say if it was resistant? How would you explain what organism it is?
It'll be anything but strep A. So it'll be strep not A.
Yeah, strep not A or non-A. Same with staph. If it's not Staph aureus, it's Staph not aureus, some other staph, right?
And the reagents too. So for what's the what's the inhibitor or the indicator for an indole test?
Okay. Yeah, what is
Those
the
always get
indicator
tricky.
for an indole? Yeah, the indole is not one we spent a lot of time on. Well,
All
first
right.
of all, what would it look like if it's positive for indole?
It'll be pink. No,
Yes,
yellow, right?
it's
What
pink.
is it
It
pink?
would have
So
like a
you mean
pink layer.
the layer will be pink,
Yes, it's like yellow with a pink layer on top.
right?
And what is the indicator for that one? It's not phenol red. The other
No,
ones that are
it's,
pink, a
it's
couple of them
the
are phenol red.
it's the Kovacs reagent.
Kovacs reagent.
Kovacs.
And I don't know how to tell you to remember that other than just try to remember it.
Yeah, I know. When we were doing it in class, I definitely missed it. I didn't remember it, but
Yeah, it's one of the
Kovacs.
more obscure ones. How do you know if something is positive for motility? How do you know that?
You have growth beyond the needle injection.
Right, growth beyond the stab line. And what structure does that mean the bacteria has? If it's motile, if it has mobility or motility, not mobile, well, it kind of mean the same thing, motility.
I mean, by the way, guys, feel free to jump in. I feel like I'm hogging all the answers. Go ahead and like try to jump in. I'm trying to kind of
You
pause
know,
a little
that's the
bit.
male versus female brain thing.
Is it?
Girls, females will generally wait till they know they have the right answer and then they'll say it.
Interesting.
Males will talk their way to the answer.
Well,
Males will, if you're raising your hand somewhere, this was
uh,
like from high school. This was like in my education classes, and it bears out. It's true. Males will raise their hand because they want to be first. Females will raise because they want to be right. Guys don't care if they're right. They just want to be first.
that's an interesting observation.
It's very
I never thought of that,
true.
but you were right.
Because I remember like a guy will have his hand up and I'll call him and he goes, well, I don't know. Well, why do you have your hand up? Because he wanted to be first. Anyway, Justina, what do you think? Oh, I forget what the question was. Oh, what structure do the bacteria have that have motility? It's not cilia. It's not.
Flagella.
It's
I'm
flagella.
over here talking and I'm muted. I'm
Oh,
sorry.
okay. I was going to say it's the same as sperm cells have. Flagella. Remember, bacteria only have flagella. They don't have the cilia or the amoeba. Those are in the. You don't have to worry about those for this lab. This lab exam. Those were in the protists. Let's see what else. Oh, what's better, a high or low TI, therapeutic index?
Low.
Anybody want to agree
High.
to disagree? You want a high TI. How do you calculate it?
That would be the patient toxic index over the patient index.
Yeah, the toxic MIC over the
toxic
over the patient MIC.
Yeah.
And the bigger the number, the better, because that means that the toxic level is further away from the patient level. Like you want to be as far away from toxicity as possible, so that, and because it's a ratio, bigger as that difference gets bigger, the result gets bigger. So the therapeutic index is higher. That makes sense. But you want a low patient MIC because you want the lowest level of treatment that's effective at controlling the organism. Like you don't want to give somebody, I don't know, 8 microliters per milligram or microgram or whatever the heck. I don't remember what the units are, but if 2 will do, right? So you want the lowest level that's effective. In antimicrobial studies, the DISC, and I always forget, is that the Kirby-Bauer? Yeah, Kirby Bauer's the
disc one,
the
right?
disc diffusion. How will you know if she shows you a picture? How will you know which is most effective?
The zone of inhibition will be the biggest.
For antimicrobial.
Well, it will be, you'll have to consult with the
Yes.
CLSI.
Nicole, that one, it's gotten me. It's gonna
I'd
get
rather
people
have to consult the
the
guidelines.
guidelines. But for disinfectant studies, bigger is better, right? On the back of the
Right.
disinfectant study page, I have bigger is better disinfectant tests, like with a big star, because I don't, because yeah, you wanna say the bigger zone is more effective, but only for disinfectant studies, right? What about quantifying microbes on a traditional streak, which is the one with the quadrants? How do
So,
you
by the way, what do we call, is that just called the, like, quadrant streak? Like,
you can
would the
call
name?
it traditional or quadrant, either one is okay.
Okay.
I
call
was
it
calling it
traditional.
a tertiary quadrant streak because
Yeah, because you only go to
it
the
goes
third.
off
Yeah.
crazy. But
Yeah,
I don't
the
know if she gives
tertiary
me,
streak. I
I
call
don't know
it
if she would
traditional
give
just
me,
because that's the way I learned it, but
Okay.
either one would be okay. Yeah, it's not, we don't use all the quadrants, we only use 3 of them. How
right,
will you quantify? Let's say that you have normal flora into the second quadrant and you have pathogen into the third quadrant. How would you quantify those?
Well, we'll say the pathogen is, who,
I think she said she had to go. I think she just had to go. She had something. Oh,
okay.
no, maybe not. She's back. She must have gotten dropped off.
All right, no, normal flora, it would be considered, so so it's like few, moderate, and what's the third option?
And
Heavy.
heavy.
Heavy. So
I always learned it
pathogen will be
light,
heavy.
moderate, and heavy, but I think few or light. Is it few that she used? Is that how she?
I think she used few. I think few.
Yeah, few,
Yeah.
moderate, heavy.
Yeah, use the word she used. I used light, moderate, and heavy, and I think that would probably be okay, but use few if
Oh, I don't know the answer
use
to
what
that.
she
I don't
used.
know what
So
few.
pathogen will be
Light
heavy
or
and
few would
normal
probably
flora will
be
be moderate then.
Right. Yeah, so remember you don't actually count there. And then what about the urine streak? How's that differ?
So that's the one where you count, you use the colony forming units.
Yes, so you want to report them, and how do you get from colonies? If you, let's say you count 40, how would you report that?
So you just multiply that by 100,000. So is it 100,000?
Just 1,000. Just by 1,000.
Okay, okay. So it'll be 40,000 colony forming units.
Yeah, you just multiply times 1,000. So they're reported to the physician as colony forming units. I was trying to see if I could pull my whiteboard up, and of course it's also missing in action. No, we'll just talk through it. So let's say you have 8 gram-negative colonies. You have 20 gram-positive colonies, and you have 15 normal flora. So 8 gram-negative. I already forgot what I said. 20 gram-positive and 15 normal flora. How would you quantify those?
Oh yeah.
How would you quantify the pathogens? That's probably how she'll do it. She caught me on one of those the other day. I'm like, oh. You
So
multiply times what?
by 1,000.
Times 1,000. So you have
So
8,000.
he said 8.
8 20 gram-positives and 15 normal flora.
What is the exception again? I need to go back on
Well,
that.
we'll get there. We'll talk. Let's see. If we have 20, so it'll be 8,000, 20,000, and 15,000. But which one is reportable? Which is there's only one that's the pathogen. Which one would that be?
The gram positive,
Yeah, because
because
unless you have more than 10 colonies, you're not, or 10 or more colonies, it's not reportable as pathogen. It's just too small of a
so
population.
how about the normal flora in this case?
The normal flora, if she says quantify the pathogen, you wouldn't report the normal flora. But let's say you had, would you reject this one for any reason?
It's two of them that are pathogens and then one that is the normal flora, right?
Yeah, except the gram negative is less than 10, so it's not
Well,
reportable.
it has to be 3 types of pathogens that are
Right.
over 10 or
Or-
over 100 colonies of normal flora
Right.
for it to be rejectable.
Exactly. So this one's not rejected because it doesn't have 3 or more pathogens, right? Usually pathogens in urine streaks, usually they're fairly, they're usually one type of microbe. It's not usually a mixed situation. But I mean, it's possible to have 2, but usually if you have 3 or more, it usually means contamination of some sort. And you don't have more than 100 normal flora, because urine's supposed to be, sterile, so there shouldn't be normal flora, right? If there's normal flora over 100, it came from improper sampling or from surrounding tissue or something. It usually means that it was contaminated. What if you had the same scenario, but you had 110 normal flora?
Then it would be rejected.
Then it would be rejected because you have more than 100. Does that make sense? Dembo, does that make sense to you?
Hold on, can you repeat that?
Yeah, so there's 2 sources of rejection are either over 100 normal flora, or you have 3 or more types of pathogens.
So it'll be a 3 or more types of pathogens if all 3 of them are over 10.
Well, except, yeah, that's right. Yeah, they'd have to all be over 10.
Including the normal flora or including another?
Not including the normal flora, just the non-normal flora, just the gram negatives, gram positives that aren't normal flora.
But what else would be a third pathogen if we eliminate?
There could be 2 different types of gram negatives or two different types of gram
Okay,
positives,
gotcha.
maybe.
If they're over 10, then you'll reject it if they're
Yes.
over 10.
That's right.
And then the other one is if the normal flora is over 100,000 colony-forming units.
That's right.
Okay.
Or over 100 colonies. Yep.
Okay, cool.
Well, we're gonna have to get off here. I'm sorry. We could just go on for hours.
Appreciate that. Thank you.
Yeah.
very
Well, I'm
much.
glad we got, if nothing else, we accomplished getting you to know that you have your lab test on
Oh
Sunday.
yeah. What would have happened was I would have procrastinated thinking it was just the lab quiz, and I'll
And
do
then
it
you would have
on
seen it.
Sunday,
Yeah.
and then I'll realize, oh damn, I have
Yeah.
this to do, and I gotta
Double
like
damn, I'd say.
study before I do it. Is a whole
Yeah.
thing. So I'm glad.
Well, and if you go back, just go back through your activity guides and the review that she did, that's posted as a PowerPoint.
Right.
Yeah, and that chart that we were working on, that should be helpful. Like you can quiz yourself off of that. I'm just looking through real quickly to see if there's anything we didn't really touch on. I think we touched on a lot of them. I don't know if we touched on all of them. But yeah, make sure you know whether something's acidic or alkaline. I don't think she asked a lot about what the enzymes are other than amylase. Oh, no. Was it amylase? Yeah, the amylase for the starch test. I think it's, that one's fair game because she's mentioned that a lot. But some of those big long ones, I wouldn't worry too much about those. Oh, the blood auger ones. What are the 3 different types of hemolysis? Make sure you know those. Somebody want to say those?
Yeah, alpha, gamma.
Alpha, beta, gamma. And which one is yellow?
Beta.
Beta. And is that complete or incomplete?
What do you mean?
Like, is it complete or partial hemolysis? If it's yellow, it's
Complete.
Complete.
it's complete. Yeah. And that annoys me because I feel like alpha should be the one that's complete because it's alpha, but beta is yellow. Make sure you say, like, the one that she showed, it looked yellow to me, but it was green. So it should have been alpha.
That happens to me all the
But
time.
but say what you see. Like, if you think it looks green, say green and alpha, right? Or if you think it looks yellow, say yellow and beta, because then she'll at least know that which color goes with which thing. Because
I always tell the difference by I say it's beta when it's like distinctively really bright.
okay, that's good. Yeah, and it is. If you see them side by side, it's usually not too bad to figure out which is which. But if they're just a single one, sometimes I think the green looks yellow or the yellow. But yeah, you're right. The yellow is more vivid. It's more, and it's that way because it's cleared the red out. It's like not muddied up with red blood cells because it's completely it's complete hemolysis. The red blood cells have been completely lysed by what's the enzyme? Sounds like the word
What do you call it?
Myelin.
Hemolysin.
hemolysin. Think about again what it means. Hemo meaning blood, lysis meaning to break down, right?
I've got my work cut out for me.
Hemolysin does hemolysis. So it's 6 through 12, right? On your test, lab 6 through 12.
Yeah, that's what she said.
Yeah, that's right. I'm just paging through real fast to make sure we didn't, like, I think we touched on a lot of things. What's the one that's this is a black precipitate masks the motility test?
The sulfur.
Yes, sulfite, good. That's the one. And what's the indicator?
Iron.
Iron. I always think about iron. Iron's black, right? A lot of it is black. So it's the one that could potentially mask the motility test because they're all done in that sim tube. Remember, there's 3 tests in one. Yeah, we had quite a conversation about that the one week, I remember. Like, if we could do them independently of each other. And we wonder about things, don't we? I think that we've touched on the highlights, but I would go back. I would say go back to your lab quizzes, especially the things that you got wrong on your lab quizzes, lab quests, I guess. And make sure you can answer the questions because you were able to use resources on those. So make sure you can answer those without your resources. And yeah, go over the review PowerPoint. I think those 2 things would be.
So on the lab exam, we can't use resources, right?
That's correct.
Okay.
Yeah. So make sure you can go back to those lab quests and answer the questions and without your resources.
Right.
All right, kiddos. Good
All
luck
right,
to
thank
you.
you.
See you on
Thank
Monday.
you.
No, will I
Take
see?
care.
Yeah, I'll see you Monday.
Bye.
Have a good weekend. Take care.
You too.
Bye, everybody.